Hello everyone. This video is about restriction enzymes. Before discussing about restriction enzymes, first we will have a look on restriction modification system of bacteria. We all know that with tail fibers, these bacteriophages attach to the bacteria and insert their viral DNA into the bacteria. After inserting, this viral DNA hijacks the bacterial DNA and prepare its own proteins into the bacteria. Due to this, lot of viral particles get produced into the bacterial cells and bacterial cells get destroyed. But just like us having immune system, bacterial cells also have a system to fight with invaders called restriction modification system. It has two components, restriction and the modification. Bacteria have restriction enzymes which cut the viral or foreign DNA into fragments in order to protect bacterial cell. But now the question is, restriction enzymes can cut viral DNA at particular recognition sites, then why do the restriction enzymes not cut the bacterial DNA? The answer is on the screen. Because bacteria can protect their own DNA by adding a methyl group to the recognition sites into the DNA. The culmination of this work was the purification of restriction enzymes, and in 1970, the first restriction enzyme, HIN2, was isolated from the bacteria Haemophilus. Now, what are restriction enzymes? As we have discussed earlier, restriction enzymes have a property of cleaving DNA molecules at or near specific sequences, and that is why they are called as molecular scissors used to cut DNA, and they belong to a large family of enzymes, nucleases. They can be classified into two types based on the site of cleavage, endonucleases and the exonucleases. Endonucleases cut polynucleotide chain of DNA in the middle, whereas exonucleases cut polynucleotide chain at the ends. Restriction enzymes cut the DNA at some specific nucleotide sequences called palindromic sequences, and these palindromic sequences have an interesting characteristic. Let's take an example of word level. 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 Reading it in one direction matches the reading in opposite direction. Just like this, palindromic sequences are. Reading of sequences in certain direction on one strand matches the reading in opposite direction of the complementary strand of DNA. Cleavage by enzymes lead to two types of DNA fragment ends, sticky ends and the blunt ends. The basic difference between these two is sticky ends are overhangs whereas blunt ends are flat. Now, there are two terms, isoscissomers and neoscissomers. When two different enzymes have same recognition sequence and same cut site, they are called isoscissomers. But if two enzymes have same recognition sequence but different cutting points, they are called neoscissomers. For example, in neoscissomers, the enzyme XMA first and SMA first have same recognition sequence, that is CCGGG. But in XMA1, the cleavage occurs after first C, and in SMA1, the cleavage occurs after third C. Now, nomenclature of restriction enzymes. Restriction enzymes are named after the bacteria from which it is isolated. Name of restriction enzymes include four terms. First capital letter comes from genus. Next to small letters comes from species name. The third letter represents the strain type. And the fourth term is a Roman number, represents order of enzyme isolated from that bacteria. There are four types of restriction enzymes. Type 1. The enzymes which comes in type 1 cleave or cut the DNA about 1000 base pairs away from the recognition sites. That is why they have different subunits for recognition, cleavage, and methylation. Now, type 2. These enzymes cut the DNA at exact position, means at the recognition sequence, not away from the recognition sequence. And most of the useful restriction enzymes are of this type. Type 3. These enzymes cut the DNA about 24 to 26 base pairs away, and they have two different subunits, one for recognition modification and another for cleavage. Type 2 S. They are somewhat like type 2, but the difference is that they cut the DNA about 20 base pairs away. Some of the basic application of restriction enzymes include gene cloning, and this is the widely used application of restriction enzymes. Next is protein expression experiments and restriction fragment length polymerization. So these are the basic application of restriction enzymes. Thank you for listening, and if you have any query or any mistake if I have done, then please let me know in the comments. Thank you.